Hello children, welcome to biology class. We are discussing about cell division, right? Yesterday we discussed about the stages, 4 stages of mitosis. Do you remember the stages? You repeat with me? Yes, prophase, metaphase, anaphase and telophase, isn't it? Today we are discussing about meiosis. So, what is the diagrammatic representation exhibited here? What is this? Yes, it is a diagrammatic representation of sperm and it is an egg, isn't it? Children, for the formation of gametes, the type or the mode of cell division is meiosis, okay? The reductional division. Did you know the significance of meiosis? Why the reductional division is required? Yes. And mitosis, it is equational division. So, for example, you just imagine in body of Homo sapien, human being, we are having 46 number of chromosomes, right? When the cells of human being divides into 2 cells, the newly formed 2 cells will be having 46 number of chromosome itself. But it is not the condition in sperm mother cell and egg mother cell. Here, what happened in reductional division is happening for the formation of gametes. Otherwise, we can't produce the same organism during the process of fertilization, isn't it? So, for that, a kind of cell division is there for the production of these haploid daughter cells, that is meiosis. Okay. So, meiosis involves 2 cell division, meiosis 1 and meiosis 2. Meiosis 1 is initiated after the parental chromosome have replicated to produce identical sister chromatids at the S phase. And this meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids of homologous chromosomes. So 4 haploid cells are formed at the end of meiosis 2. So, which are the events? Meiosis 1 and meiosis 2. But in meiosis 1, at prophase 1, again 5 steps are there. That is leptotene, zygotene, pachytene, diplotene and diakinesis. So, first of all, let's discuss about meiosis 1, prophase 1, the 5 stages. So, the first stage is leptotene. So children, keep it in mind. In meiosis 1, prophase 1, the first stage is leptotene. During this stage, the chromosomes become gradually visible under the light microscope. Then the compaction of chromosomes continues throughout leptotene. And this is followed by the second stage, that is zygotene. And what is the speciality of zygotene? During this stage, chromosome starts pairing. See here, children, chromosome starts pairing clear together, and this process of association is called synapsis. Okay, such paired chromosomes are called homologous chromosomes. So here you can see homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of a complex structure called synaptonymal complex, and the complex formed by a pair of synapsed homologous chromosomes are called bivalent or tetrads. See here, we can see tetrads here, right? However, these are more clearly visible at the next stage. The first 2 stages of prophase 1 are relatively short-lived compared to the next stage, and that stage is pachytene. During this stage, you know what happens? The 4 chromatids of each bivalent chromosomes become distinct and clearly appears as tetrads. Here you can see a different color is given to the sister chromatids for the identification. So this stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between 2 homologous chromosomes. Clear? So I will show you one more diagram. See, This is diploid. In this patchogen here in this diagram, it is clear. Non-sister chromatids given color, and for sister chromatids another color is given. Clear, children? So this crossing over is also an enzyme mediated process, okay, and the enzyme involved is called recombinase. And crossing over leads to recombination of genetic material on the two chromosomes. This recombination between homologous chromosomes is completed by the end of patchogen, leaving the chromosomes linked at the sites of crossing over. Children, this recombination is the reason for variation, isn't it? Then diploid, next stage. The beginning of diploid is recognized by the dissolution of the synaptonymal complex and tendency of the recombined homologous chromosomes of the bivalents to separate from each other, except at the site of crossing over. See in different color I have given. Okay, so except at this region, the separation takes place. Okay, so here you can see an X-shaped structure. Can you see X-shaped structure here? X-shaped, alphabet X, and it is termed as chiasmata. Okay, chiasmata. So here you can see chiasma, and together it is called chiasmata. Spell it as CHIASMATA. In oocytes of some vertebrates, diploid can last for months or years, isn't it? Then next one is the last stage, that is diakinesis. This is marked by termination of chiasmata during the phase of the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents a transition of metaphase. Children, so that is diakinesis. So that is about prophase 1. Okay, so only prophase 1 we completed now. And what is the next stage in meiosis, children? Yes, metaphase 1. See, this is metaphase 1. Here the bivalent chromosomes again on the equatorial plate. Okay, so the microtubules from the opposite pole of the spindle attach to the kinetochore of homologous chromosome. So here you can see the attachment of spindle fibers. Then it is followed by, yes, anaphase 1. The homologous chromosome separates while sister chromatids remain associated at the centromere. Isn't it? Here you can see the attachment of sister chromatids. Then telophase 1, the nuclear membrane and the nucleolus reappear, cytokinesis follows, and this is called a dyad of cell. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interface of nucleus. The stage between the two meiotic divisions is called interkinesis and is generally short-lived. That means there is no replication of DNA during interkinesis. Interkinesis is followed by prophase 2, metaphase 2, anaphase 2 and telophase 2. Clear? So that is about meiosis 1. Then what happens during meiosis 2? So here now we can see 2 daughter cells, and from these 2 daughter cells 4 daughter cells will be formed, and the formed daughter cells will be having half the number of chromosomes, isn't it? If the animal is having 12 number of chromosomes, how many numbers will be here? Yes, 6. If 18 number of chromosomes are here at the starting, yes, here 9 chromosomes, like that. So being Homo sapiens, we are having 46 number of chromosomes. So our sperm and egg are having 23 number of chromosomes, isn't it? So 23 chromosome in sperm plus 23 chromosome in egg fuses during the process of fertilization, and 46 numbered zygote is formed. Here, you got the idea, children? Yes, that's about meiosis 2. And what are the stages of meiosis 2? Same as that of metaphase. but we have to mention prophase 2, metaphase 2, anaphase 2, telophase 2, but the steps are same. So let's discuss prophase 2. Meiosis 2 initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. So the nuclear membrane disappears by the end of prophase 2 and the chromosomes again become compact. Then about metaphase 2. Metaphase 2, here you can see metaphase 2. At this stage the chromosomes align at the equator. And the microtubules from opposite poles of the spindle get attached to the kinetochore. And what happens during anaphase 2? Here it will be split. It begins the simultaneous splitting of the centromere of each chromosomes and what happens, allowing them to move towards opposite poles. And finally during telophase 2, what happens? The meiosis ends with telophase and 4 daughter cells are formed. Two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad. 4 cells, tetrad cells are there. So what are the significance of meiosis, children? It helps for the formation of gametes, isn't it, for the animals, for sexual reproduction, for the formation of gametes. It is required. Not only that, during prophase 1, an important process is taking place. What is that? Recombination. Recombination was the not result, it's a reason. Recombination is a reason for variation. Variation is there in our world. Otherwise, we can't survive in the changed nature. We know that mutation is taking place. So each and every organism has to be changed. The variation must be there among species, and it helps for the formation of new species too. So this is a significance. It is very important. I hope you got the idea. You read and read a lot. Okay, you read the textbook thoroughly. 5 times you read the steps. It is very important, children. And study well. Okay, thank you, children.